now we have to go over essentially how the neuron conducts that electrical conductivity from one neuron, from the neuron from the cell body down the axon, which you'll learn about, and how that's going to get transmitted over to its intended organ, whether it's going to be a muscle, a gland, or another neuron. So with that, what we do need to understand is to go over some of the fundamental concepts, of which we're going to talk about the membrane potential. What's going to give rise to this electrical voltage difference? And that's going to be related to the ions and the stuff that's found inside the cell in the intracellular fluid versus that found in the extracellular fluid. So the term that we're going to be using is called resting membrane potential. This is the neuron at rest, where it's awaiting sensory information. Not doing anything right now. It's completely sedentary, for lack of better terms. And then we're going to look at things that result in changes in the membrane potential. We can depolarize it, repolarize it, and then you'll see also hyperpolarization, and we'll understand the changes that are taking place in the resting membrane potential. So you'll see the terms are basically described in here. I'll get into it as we move into the membrane potential, but keep in mind that this is the state. This is the energy that's found in the state of that cell at the time, which is a form of potential energy. So let's take a look at the key players that are going to be involved. Aside from sodium, aside from potassium, we're going to talk about the things that carry them in and out of the plasma membrane. And as you answered this from your quiz question, these are the channels that are involved. So these channels allow ions to move through, of which the channels, as they move through, as we start getting ions moving out, we get a potassium, a positive ion moving out, that's going to cause changes in that membrane potential, which we'll describe later on. Nonetheless, ion channels allow ions to move down their concentration gradient. We move it down from a high concentration, so if we see here a high concentration, down to a low concentration, and here I'm referring to potassium. It's going to move from a high concentration out to the low concentration. We'll see how that's going to govern the movement of ions from inside to out for our potassium. And then vice versa for sodium, moving down those channels from outside in. And then not only that, these channels aren't always open. And you'll learn about certain channels that will either be open or not open. Well, I mean not open, but governed by gates. And so here you'll see the types of channels, gated and non-gated. So our non-gated channels are channels that are constantly open. And the best case example that we can talk about are leakage channels. So when we talk about leaky potassium channels, if you understand the term leaky potassium channels, you know that these channels are constantly open. And we know that the concentration is high on the inside, while it's very low on the outside. And so that would allow potassium to constantly move out of the cell through these leaky channels. And when we talk about leaky channels, we have more potassium leaky channels than we have leaky sodium channels. So that we'll talk about in just a little bit. Now, the question is, how do we get potassium back into the cell? And that we'll take a look at, and we'll answer in just a little bit. The next thing we're going to take a look at are gated channels, in which we have channels that open and close based on a specific stimulus. And the three stimuli that you see here are voltage gated, ligand gated, and mechanically gated, of which the first two we're going to be talking about heavily. Voltage gated. Voltage gated requires a change in that membrane potential. So there has to be an electrical change that elicits these channels to open up. The next one, ligand-gated channels, are channels that open in response to something binding to it. So ligand, remember that term ligand, is something that binds to, in this case, would be a receptor. It binds to that ion channel, okay? And give you an example would be a hormone like insulin, for instance, any neurotransmitters, or other ions that bind to. So they can bind to ligand-gated channel and allow sodium to flow through. Otherwise, if that ligand is not bound, then this channel remains closed. And in most cases, at least what we'll talk about, are neurotransmitters. So neurotransmitters that are released have to bind open to these channels to allow them to open up and allow sodium to move in. And again, when sodium moves in, that's going to change the membrane potential of the cell membrane. And then lastly, mechanically gated. This we'll talk about in our sensory lecture. In order to open up that type of channel, it requires an actual mechanical force to open them up. So any changes to the cell membrane causes these to open up. Signal gated, this one isn't really the best example on here. So I see voltage gated, ligand gated, signal gated channel. That's not really the best example, so I'm going to X that out. But what that really is, if you want to know what signal gated is, there's a signal that happens intracellularly. So something on the inside causes it to open up. So that is still considered a ligand gated channel because it requires something to bind to the receptor to open it. So now that you know the key players, you know the terms that we're going to get into, let's knock off those terms. And while we do that, we do have to go over some basics. And so here we can review electrical energy. And I'm not going to get too much into the physics of things. But as we see, there's a relationship between voltage and current. There's going to be a relationship with voltage as well as the current of ions moving through. So we can use the example of a hydraulic system. And what I'm showing you here is high potential energy. We have a hydraulic system where we have two columns filled with water, and that's connected by a single pipe in between. And in the center, we have a valve that we can open and close to govern how much stuff is going to move from one direction to the other, or from one side to the other. So seeing this hydraulic system, you see that one side has a higher water level, and the other side has a lower water level. And we know that the area that has the highest water level has the highest amount of potential energy. Why? Because there's a lot of osmotic pressure, hydrostatic pressure that's pushing down in this area. So we move from a direction of high potential over down to low potential. So in this case, what we're going to be able to see in our cell is we're going to see an example of high potential moving down to low potential. And that's going to govern the movement of this electrical current that's going to come in the form of ions. And what represents that valve in our hydraulic system is going to be represented by this resistance, which is going to be the cell membrane. That valve is going to govern the ions moving in and out. And as those ions move in, you're going to see that the membrane potentials, the potential that we're going to be able to see, is going to change. And then looking at our next slide, this is really just a review. So this I don't want to go too much into, but we know from our cellular physiology lecture from last Tuesday, we know that the extracellular fluid is higher in sodium, higher in potassium in the intracellular side. Not only do we have a lot of potassium on the inside, but we've got a lot of amino acids. We've got a lot of phosphate ions. And where do you think those phosphate ions come from? You might remember we talked about this on Thursday. Our energy currency, ATP.
Oh yeah, ATP.
Yeah, ATP. Once ATP breaks off, it's going to release, it's going to turn to ADP, and it's got a phosphate ion on there. Those phosphate ions were usually negative. On top of that, we're also going to be able to see a bunch of proteins in the cell, and a bunch of organelles, huge structures that lend itself to the membrane potential. So let's take a look at exactly what I mean by the membrane potential. And what I mean by the membrane potential is the difference in the charges, the difference in the charges on the inside and out of the cell membrane. So if you were to take a look at the distribution of those ions, you would see that for the most part, far away from the neuron, the concentration, the difference of ions, the charges of the ions are going to be about the same. But the closer you get to the cell membrane, you're going to see that there's a certain bias to it where the inside is going to be slightly more negative than the outside. And we're going to talk about what's going to contribute to those. But the difference is the membrane potential is the difference between the outside versus the inside. And in neurons, you're going to see it's going to range from -40 to -90. But the number that we're going to be working with here is -70 millivolts in our classic neurons. And so we have a way of actually measuring that membrane potential, where we take very sensitive electrical equipment, a voltmeter, and what we're going to do is we're going to take one probe that's going to inject underneath the axon, or right underneath the cell membrane, right below. And the reference electrode is going to be the extracellular fluid surrounding on the outside. So this is going to be electrically neutral. And then when we measure the inside, we'll note that the difference in between, it's going to say that the inside is going to be slightly negative. And if you were to measure it, it would be about -70 millivolts. And ever looking at cellular physiology when they're testing these, or when you're testing neurons, you're doing this all the time. You've got a probe one out here, and you've got another probe that you've injected right underneath the axon to measure that difference. So the question is, why is it negative on the inside? Well, let's take a look. The negative resting membrane potential, and what we're going to take a look at here is just a box of some sort, or some vessel, and right in between you're going to see the semi-permeable membrane. Very similar to that YouTube example that I showed you earlier. And on one side you're going to see, if we were to take a look at the charges of things, we're going to see our potassium ions, which are going to have a positive charge. And then all these M negatives are the other impermeable anions. And what exactly are those impermeable anions? Phosphate, ions, proteins, stuff like that. So all the organelles, these are the big things that are in here. And you'll note that the semi-permeable membrane only allows certain ions to move through. And in these cases, these are going to be channels, particularly channels that remain open for potassium, or leaky potassium channels. That's the only thing that can move through because these channels are always open. So looking at this, you'll see that the concentration of potassium is highest in the cytoplasm. And you'll see on the extracellular fluid, there'll be a little bit of potassium here and there, but considerably far less potassium on the extracellular fluid, as I mentioned in this slide right here, higher potassium on the inside. So my question to you is, what would be the direction of travel? We go from high concentration to low concentration. So in this case, which direction is the potassium moving through?
To the right.
To the right, out into the ECF, right? And now what happens if we get our potassium ions to move out? Think about it here. Here you see a bunch of M- and a bunch of K+, or potassium positive ions and negative ions. If I get positive ions to move out, what is that going to do for the intracellular fluid?
It becomes negative.
A little bit more negative. So now it's more negative on the cytosol. We have the concentration gradient pushing potassium out. But we're stuck with something where it's slightly negative on the inside and you've got a positive ion. So while I have the concentration gradient moving out, I have potassium wanting to get slightly pulled back. Okay? Because it's a positive ion and it's negative on the inside. Now those channels might not necessarily let potassium out back in, but it creates this gradient. So you've got a bunch of potassium close by and you've got a negativity on the inside. So negative on the inside, positive on the outside. Also think about what happens once you start getting more positive ions on the outside. You've got a positive ion trying to leave down its concentration gradient, but at the same time, it's also positive over here. So you've got positive versus positive. So there's less of a push, less of a drive. In other words, while you have a concentration gradient moving out to the right, you have the membrane potential, the negative side wanting to limit the amount of potassium moving out. And there's always this balance during rest. Remember, this is still resting membrane potential. So taking a look at that, this is just another slide basically illustrating the same thing, where potassium are mixed in with our anions. The concentration gradient, represented by green, is going to want to drive potassium out of the cell, while the electrical gradient, or the membrane potential gradient, wants to pull our positive ion back into the negative intracellular fluid, or at least the negative intracellular side. And the movement of potassium will keep moving until an equilibrium is found. That means that not all the potassium will move on the way out. You'll get potassium that will be in equilibrium on this side, and the remaining potassium on that side, so that it remains at this negative resting membrane potential. And we'll talk about other contributors as to why it remains negative, two of which I already gave you. Yeah. One, potassium moving out the leaky channels. The other are the negative ions. You'll see that on the next slide over, so don't worry about writing that down. Okay. So this represents our equilibrium. And now we're tied in. Something else I want to point out is all we've talked about right now are positive ions, or specifically our potassium ions. This is the equilibrium that's found specifically with potassium ions. And as I mentioned earlier, when we talked about the movement of potassium out, we would have to understand this whole experiment that they were able to run a long time ago when they were trying to understand membrane potentials in cells. And what they were able to do is take a cell, and they were given the ability to tweak the membrane potential themselves. And they can basically send electrical currents that would change the membrane potential. And that would allow us to understand the movement of specific ions. They can literally turn on a sodium channel, and they can turn on potassium channels. So in this experiment, what they were able to do by turning on only the potassium channels, they can see where the equilibrium would be. So if only potassium channels and every other channel were turned off, well, what would happen is you would see the membrane potential about negative 90. And that means if you're to take a look at this example earlier, that means if you're going to get potassium out and then it stabilizes with the concentration gradient and the membrane potential, that means that the difference from the outside and the inside is negative 90. Now, turn everything back off. Get it back to where you started off from the very beginning, and just work with the sodium channels. And turn on just the sodium channels. And think about the same concept. Here you're going to have a bunch of sodium on the outside. You got a little sodium on the inside. Now, what direction is sodium going to move in based on the concentration gradient?
From right to left.
To the left, right? So it would move intracellularly, correct? Mm-hmm. Now, from a membrane potential perspective, it's going to be slightly negative on the inside because of this distribution that you saw earlier, where you have these big ions. So from a membrane potential, if it's negative 70 on the inside, which direction do you think a positive ion wants to move? Do they want to move towards the positive side, or they want to move towards the negative side? Or the negative side. So these sodium ions also want to move intracellularly. So there's a drive for both the concentration gradient and the membrane potential for sodium ions. And again, we'll revisit this on Thursday when we first start class. Now, looking at this, if they were to turn on all the sodium channels, you see that we have positive ions wanting to be driven into the cell. Now, my question is, if it's normally negative due to the negative resting membrane potential that we were able to establish on these slides earlier, If you have positive ions moving into an intracellular environment, what is that going to do for the intracellular side?
Make it positive.
Make it more positive. And where is that equilibrium? If you only turned on the sodium channels, it would make the inside more positive, and the membrane potential in that case for sodium would be about positive 65, 70. You don't necessarily need to know these because you know that when we look at the sum of all potassium channels and the sum of all sodium channels at rest, there's a balance of each of both. And the key thing about our potassium channels, and I want to kind of jump into this topic here because I'm going to wrap it back to my last one, is that we have more leaky potassium channels than we do have sodium channels, at least leaky potassium sodium channels. So most of those leaky channels are potassium. So that means potassium is more free to move than sodium channels at rest. So if this was our equilibrium point for potassium, and this was our equilibrium point for sodium, and we know that we've got a lot more leaky potassium channels, where do you think our membrane potential would be? Would it be closer to potassium or would it be closer to sodium? It would be closer to potassium because we have more potassium channels. If you have more potassium channels, that means more potassium is moving. And again, at that example I said earlier, if we're only mostly seeing potassium move, we would probably be closer to negative 90. But because sodium is involved, and that's on the positive side, still affect a little bit of effect of sodium. So the balance that comes out to negative 70. Again, that slide I mentioned earlier, negative 70 millivolts. That one goes into much more. If you were to go into the book, they start to go into the Nernst equation. They go into looking at equilibrium points. But that's really the sum of it. The key point I'm trying to get at is potassium is one of the main reasons why the intracellular membrane potential is slightly negative. So we see more potassium channels. We see the unequal distribution of potassiums on the inside versus the outside. And potassium wants to leave. So let's take a look at that. If this is the intracellular fluid, we have positive ions moving out. You already said, what is that going to do for the intracellular side? A positive ion moving out.
Make it negative.
It's going to make it more negative. So that's the first factor. That's the first main factor for the negative resting membrane potential. The second is the inability of most anions to leave the cell. These were the phosphate ions. These were the big proteins, the amino acids, that are typically negative in nature. And they don't go anywhere. And that's also going to make it negative. You have anions on the inside. That's going to make it more negative on the inside as well. The third contributing factor are the other thing that we didn't talk about, our sodium-potassium pumps. And remember, what our sodium-potassium pumps do is they're going to actively pump ions against their concentration gradient. And when I mean active, what molecule are they using to fuel this? ATP. So it utilizes ATP through the removal of that phosphate ion. We're going to be releasing the kinetic energy from that ATP to fuel those pumps to pump potassium into the cell and pump sodium back out of the cell against their concentration gradients. But if you're going to look at the distribution of those charged ions, take a look. 3 positive charges out, 2 positive charges in. Is that equal?
No.
No. So if I'm getting less charges going in, that should make it more negative, shouldn't it? What questions do we have about that?
You said more positive charges going in, right?
I'm sorry, more positive charges going out.
Oh.
And less positive charges going in. So there's more positive going out, making the inside slightly more negative. So those are the 3 contributing factors, of which potassium, the leaky potassium channels, is what causes the biggest one. So looking at this, now that we know the negative resting membrane potential, I got a video here that's going to help summarize all that for you, what's happening. So it's about 4 minutes.
Unstimulated, neurons maintain a constant electrical difference or potential across their cell membranes. This potential, called resting potential, is always negative inside the cell. It ranges from -40 to -90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative depending on the stimulus. If potential is made sufficiently less negative, it reaches a level called threshold, and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell restores its negative resting potential. The cell membrane of a neuron encloses cytoplasm with various ions dissolved in it. The neuron itself is immersed in a salt solution, the extracellular fluid. The ions of the cytoplasm consist mainly of positively charged potassium ions and large negatively charged organic molecules, such as proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up cell membranes, they must travel through channel proteins that extend through the membrane. In an unstimulated neuron, only potassium ions can cross the membrane, traveling through specific proteins called potassium channels. Although sodium channels are also present, in unstimulated neurons, they remain closed. Since only potassium ions can cross the membrane, and potassium ions are more concentrated inside the cell, they diffuse out of the cell, leaving the large negatively charged organic ions behind. As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside. At some point, the diffusion of potassium ions out of the neuron due to concentration differences is balanced by the electrical attraction tending to pull them back inside. This is the point at which neurons reach resting potential. Reaching resting potential in this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of the potassium ions initially inside a neuron must leave to create a resting potential of -60 millivolts. But it is action potentials, not resting potentials, that carry information through a nervous system.
That we'll talk about on Thursday. Okay, great. So now that we've talked about the negative resting membrane potential, the membrane potential of a neuron at rest, we know that it's slightly negative on the inside, positive on the outside. And any changes to the membrane potential can result in changes that can be elicited that could cause those action potentials, which we'll be describing later. In other words, the membrane potential exhibits this excitability, its ability to be changed by the movement of ions. So the thing that we need to look at that's most excitable is all related to where the channels are. We know that as ions move in and out, that's going to cause changes in that particular location. So in other words, excitability is related to the concentration of receptors, if you want to get real technical about it. And where the highest area where we have, or the highest concentration of channels are found in the axon hillock, this area right along that starts off the axon. This is between the cell body and the axon itself. So to think of something, if you were to take a look at the match wood itself burns. It exhibits chemical properties that allow it to burn. but the area that's most sensitive is the match head. And this axon hillock is the match head because it's the area with the most concentration of, I guess in the match head is more volatile things. In this case, this is where you would see all those channels. So one thing when we measure the membrane potential, if we were to take a look at it from a graph, and I don't need to draw the graph because you'll see the graph on the next slide, we can measure the membrane potential, which is at rest, negative 70 millivolts. And any changes to the membrane potential can be explained by these three terms: depolarization, repolarization, and hyperpolarization. What we're not going to get into just yet is what's going to govern the movement of ions. But pretty much based on what we've already learned, you'll be able to surmise which is going to cause which. So for now, let's just take a look at the changes in the membrane potential. Right now, you'll see that this is going to exhibit a certain polarity. If you look at all these terms, they all refer to polarization. Meaning you have a certain pole. You have a negative pole, which is the negative intracellular side, and you have a positive side, the extracellular pole. So it's clear that this negative 70 elicits a difference between the intracellular side versus the extracellular side. And a depolarization is a decrease in the polarity, meaning we're approaching, think of approaching 0 millivolts. The closer we move towards 0 millivolts is a depolarization, meaning that polarity, we go from negative 70, negative 60, negative 50, negative 40, all the way negative 20, that polarity is being lost. Repolarization is heading back to, we're reestablishing that negative membrane potential. Up until the point that we get to that resting membrane potential. Repolarization is all the way from out here, back down, back to negative 70. And any hyperpolarization, any repolarization that results in a decrease below the resting membrane potential is going to be called hyperpolarization. So these are the big changes that are taking place. So thinking about the depolarization, repolarization, and hyperpolarization, let's take a look. Give me a second. And okay, let's kind of help point out what's going to cause that. If to look at our cell, and we know at a negative 70 on the inside, we have an example that we talked about earlier. What would cause the inside to become more positive, to approach 0? We talked about two ions, sodium high on the outside, potassium high on the inside. Now, which of these two, these are the only two ions that we're talking about in this case. I want you guys to think about this. Which of these ions is going to be related to depolarization that's going to decrease the polarity? In other words, make it more positive. Is it the positive ion going into the cell, or is the positive ion moving out of the cell?
Sodium going in.
Which one?
Sodium going in.
Sodium going in, right? So a positive ion going into the cell, if it were negative 70 and I have positive charges going in, that's going to start approaching 0, hence depolarization. So depolarization is going to be due to sodium. Now let's look at repolarization. What's going to cause the reestablishing of that negative resting membrane potential? Is it going to be more sodium going in, or is it going to be positive potassium ion moving out? Positive potassium moving out. So that repolarization would be positive potassium. And lastly, hyperpolarization. What would get us to be more negative than what we already are? Is that sodium, or is that going to be potassium? Potassium, right? So this, again, is going to be related to potassium. So now that we know that, now that we know which is going to be involved, and again, that's kind of a simplification of things, let's take a look at the next slide. That's going to talk about graded potentials, where the idea behind, and just like we were describing earlier, the movement of ions is what's governing the changes in the graded potential. What's causing those ions moving in and out? How do ions move in and out of the cell membrane? The channels. So in this case, the graded potentials, the changes in the membrane potential, are due to the opening of channels, or channels that are open that allow those ions to move through in the first place. So we're going to talk about, going back to an earlier slide, which of these channels are going to be opened. We've talked about leaky potassium channels, which are always open. Now we have to talk about how we can govern that excitability based on those channels opening. That's going to cause changes to the membrane potential, of which we have voltage gated and ligand gated. In our graded potential, what happens when one sodium ion channel opens up? We get a positive ion going in, and that's going to cause a little blip in the membrane potential. So looking back at this blip, it's going to go up a little bit, and then it's going to come right back down. So thinking about this latter portion, what brings it back down, what's going to restore? Which of the things that move ions in and out of the cell is going to restore our membrane potential? Which of the channels? I'll give you a clue. It's not a channel. It requires a little energy. It's going to send ions back against their concentration gradient.
Diffusion?
It's our sodium-potassium pump. So that's going to restore it back. And I'll talk about that in one of the slides. So in these cases, when you open up one channel, it's going to cause a little blip, and then it's going to restore it back. That channel is going to close, and then it's going to restore it back to that membrane potential. Now, if we get 5 sodium channels, and this is not the exact number, but if I were to open up more sodium channels, I'll know I'll get a bigger blip. Why? Because I have more positive ions moving into the cell, causing more of a depolarization. And eventually those channels close, and then we're going to repolarize. And that repolarization, again, is due to the ions being reestablished back to their start. So looking at that, noticing that when in a cell membrane, if I have channels that open, that's going to mean that if I have more positive ions moving into this area right here is going to be depolarized, meaning it's going to be approaching 0 millivolts. And then again, if we're going to look at the concentration, or the concentration of these ions in that area, that means this area is going to be a little bit more positive, but there's going to be a lesser concentration of positive ions over here, and a lesser and lesser and lesser. Meaning the location, this area is a particularly hot zone. It's been excitable. So that, due to the higher concentration of positive ions in this area, is going to cause this area to be excited a little bit. Actually, quite a bit. So in other words, those ions spread to the nearest membranes, and it's going to cause that area to be more excited. So while if this becomes, let's say, 20 millivolts over here, positive 20 millivolts over here, maybe over here it's going to be positive 10. Maybe over here it's going to be it's going to be 0. Over here, even further away, it's going to be back to negative 70. So the strength of the depolarization is going to be related to the number of channels that are open. As I mentioned here, if this were one channel, little blip. This one is a bigger blip, so you're going to get more channels open and therefore a bigger depolarization. If you were to take a look at hyperpolarization, hyperpolarization we mentioned due to the potassium ions. If I were to open up potassium ion channels, we wouldn't see a depolarization, we would see a repolarization. The same blip. If I opened up a potassium channel, I'll get a little blip and it restores it back. If I open up 2, or let's say 5 potassium channels, I'll get an even bigger blip, a bigger hyperpolarization, again, below resting membrane potential. I'll get a blip and then it'll restore it back. And all of that is tied to these ion channels that we're opening up, as I mentioned. So what you're looking at here is the effects. This is the first effect called summation. And summation is really the additive effect. And the way we can look at the additive effect is the adding graded potentials. Meaning, if I were to get receptors that allow ions to move through, you'll be able to see that these might be able to add up. So if I get a couple channels to open and then they close, and then I get another couple channels to open, you can get an additive effect that will add up. And this is what you can see over here, where you see stimulus 1 over here that opens up these channels, and stimulus 2 that opens up these channels. So here you can see a depolarization. You've got some sodium ions moving in, and then the second stimulus elicits another depolarization, where it creates an additive effect. Otherwise, what you would have expected to look like is this on its own, and then this on its own. But because they're happening close to each other, this is the this is the additive effect.
So it's 2 sodium channels being open, right?
Pretty much. Or in more technical terms, a certain number of sodium channels opening here, and a certain amount of sodium channels opening over here. So to help tie this in, what's causing those sodium channels to open, which we'll talk about at the end of Thursday's lecture, are the neurotransmitters that are going in binding. These are the ligands that are binding to these sodium channels, allowing them to open. So help tie that in. When you get neurotransmitters that are binding from the prior axon terminal, neurotransmitters bind in, allow sodium channels to open up. And as more neurotransmitters bind to these ligand-gated channels, then you have bigger graded potentials. And again, the magnitude of that graded potential, the height of that graded potential, is all governed by the number of channels that have been opened, how you've opened those channels up. So now we have to talk about, since we're going to talk about those ligand-gated channels, we'll talk about them at the end of Thursday. We do have to talk about other types of channels that will open up. As I mentioned in that slide that I just referenced, ligand-gated channels weren't the only ones. We have voltage-gated channels. And the stimulus for these voltage-gated channels are the changes in the resting membrane potential. And we've got 2 types. We've got sodium and we've got potassium voltage-gated channels. And looking at the structure of a sodium channel, if this were the cell membrane, this is what the channel will look like, you'll actually have 2 gates. One called the activation gate, which is up here, and another inactivation gate located down over here. One on the intracellular side and one on the extracellular side. And at rest, where we have a negative resting membrane potential, our voltage-gated channels are closed. They don't move. So that's our resting state. And at that resting state, our net movement of sodium through the voltage-gated sodium channels is 0, meaning no sodium moves through. When it's opened, we're going to have sodium moving through down its concentration gradient. So it's going to move down its concentration gradient. When it becomes inactivated by the inactive gate, that stops sodium moving back in again. And that will not cause any more changes in the membrane potential due to sodium. Now the other one is potassium, where potassium gates are actually simpler. It's just the same thing, but you're going to have a channel, and there's only one gate. It's closed at negative 70 millivolts, and it's going to open. And the thing about these is they open much slower than they do our voltage-gated channels. So these ones slam open real fast, and they allow sodium to rush in. These take their time. Once the voltage changes and it allows it to open up, then it opens up and allows potassium to move out of the cell. And then it takes some time for that thing to close. So let's take a look on the next slide to see what that voltage-gated sodium channel looks like. Here you can see at rest, you don't see any sodium moving in and out because the activation gate is closed. The activation gate opens here, so it allows sodium to rush in, up until the point that that inactivation gate closes. Activation gate here is open, inactivation gate is closed, still prohibiting sodium from flowing through.
that mean if there's an equilibrium between the inside and the outside?
It just means that the channel doesn't operate anymore because it reached a certain membrane potential. So in order for it to open up, it has to have a difference in the positive side versus the negative side. So the membrane potential is going to govern whether these are open. And they open up at certain voltages. And the question is, what voltages do they open? Well, let's take a look. This piece right here we're going to take a look at. This is, and I know this bit is a little complex because there's a lot going on, but we can start off by looking at the different phases of our ion channels, our voltage-gated channels opening up. So here you can see our resting state, and a resting state of a membrane potential of negative 70 millivolts. Here you'll see all the gated sodium and potassium channels are closed, and we're back at negative 70. So when the membrane potential of an axon reaches threshold, and that threshold we'll talk about a little bit later on, is what's going to happen is the sodium is going to move in. So we're at negative 70. We have sodium opening up, the sodium channels opening up, and that's allowing sodium to move in. So my question is, are we depolarizing or repolarizing? Depolarizing. So we expect an upswing in the amount of sodium that opens up. And what's happening as the membrane potential is starting to depolarize, that's going to start opening up more voltage-gated channels. But let's not get there just yet. So what happens when we have a certain amount of, and I got to get rid of this slide because I'm not a big fan of it. I want to just draw just a blip. Okay, and this right here is negative 70. We're going to bring it up to negative 60. So negative 60, we're going to cause a little blip because we've opened up a voltage-gated sodium channel. That's going to open up, it's going to allow sodium to come up. And then what's going to happen afterwards, after that sodium channel is closed, let's turn that channel off, and we're going to repolarize. And the repolarize is due to the activation of our our potassium channels. And that's going to cause a repolarization. So positive sodium ion going in, and then potassium going back out. So depolarization, repolarization. Now, this piece of where we're going to get into, that we're going to just kind of get into on Thursday, this is the example of a graded potential, where the number of sodium channels that open will govern how big of a magnitude this is. The magic number that we're trying to get to, right? So if this is a graded potential and this is just a change in the membrane potential, this is that neuron just getting a little excitable. It just says, oh, now I'm getting really excitable. But there's a point at the membrane potential that elicits a major change. And that change is the number of our voltage-gated sodium channels. That number that you're going to see on Thursday is negative 55. Once we reach negative 55 millivolts, a bunch of voltage-gated sodium channels will open. And if you have a bunch of voltage-gated sodium channels open up, what's that going to do for the inside of the cell?
Make it more positive.
Make it much more positive. It's going to elicit a huge change. And that is a graded potential that passes. It completely surpasses negative 55 millivolts. And now you're going to get a huge rush of sodium channels going in, and it's going to cause this, which is going to be an action potential, which we'll learn on Thursday.
That's the hot spot you're talking about?
It's the hot spot. And the hot spot is the axon hillock, where if we get sodium channels to start to open up a little bit more, and that causes more of a depolarization to take place, that's going to activate a bunch of voltage-gated sodium channels in that area to open up. So it's just like this. If I were to use that match example, I have a match and I've got a flame. And the flame represents the changes in the membrane potential that can occur. And I know that probably isn't the best example. But if I were to take that fire and bring it close to that match head representing the axon hillock, it can come in and cause that, the heat can come in and cause changes in the temperature of that match head that might get close to that trigger point where it's about to burst, but it doesn't. But I get to a certain threshold of bringing in much kinetic energy, much heat up to that match, and once I reach that threshold, then the whole match lights up. That whole match head lights up, and it's going to continually burn and make its way down the length of that match.
It's like a roller coaster, right?
Yeah.
So if you reach to the top,
I'm sorry, one more time.
if you reach to the top of the axon hillock, you reach the hot spot.
Yes, if you reach a certain amount of ions reaching the axon hillock.
And
is
then
what we're
it
getting
really
at.
goes down to a straight path.
Yes, and that's the action potential. So the graded potential are just the changes happening in the areas that are receiving the information, which would be the dendrites. The dendrites are receiving, they're receiving the neurotransmitter. Again, something we'll talk about Thursday. They're receiving the neurotransmitter and they're opening up those channels. And eventually when we open up enough sodium channels, we're going to depolarize up to -55. So in other words, graded potentials lead up to, we can start to add up these graded potentials. We can keep adding up these graded potentials up to the point that we get to threshold. And that's going to differentiate. Once we reach threshold, that's going to create an action potential, which is that match completely lit up. Everything else before is just close enough.
Is that like excitability?
Yes, all of this is related to excitability, but that axon, if you remember from that video showing you that signal traveling down the axon, that's what we'll learn on Thursday. That's the action potential. That's what causes a neuron to fire a signal out. Everything else here represents graded potentials that can lead to threshold. Okay, so that's what's in stock for Thursday. So now as I mentioned earlier, I mentioned all these things about channels. I want to leave you with a couple videos. Because as I mentioned earlier, diseases can be tied into the breakdown of channels. And it might not just be a mutation or a genetic mutation of any kind. It can be a toxin, just like we learned about tetanus. And one of the toxins is called fugu, pufferfish. Does anybody know anything about fugu?
Yeah.
Anybody remember watching the Simpsons episode where Homer eats fugu?
Does it have to be like a 10-year student to
You do.
actually make, to serve it?
Yeah, so these chefs are well trained. This happens particularly in Japan where they like to eat raw fugu, which is our pufferfish. And this pufferfish has all these poisonous glands all throughout the body as a defense mechanism. And the toxin is called tetrodotoxin. And what it does is, and I'll show you in the next slide over, it blocks depolarization via inactivation of the voltage-gated potassium channels. Meaning, going back to our earlier slides, if we turn off those voltage-gated channels, we can't get a neuron to reach threshold. It'll get up to threshold, and that tetrodotoxin comes in and completely blocks those channels. So you just get this, and then it turns off. Remember, if we don't reach threshold, that neuron never fires a signal. And if that intended target for that neuron is a muscle or it's a gland, it prevents that muscle from contracting. It prevents certain smooth muscles from working.
So would that be like a decrease in potassium, causing like a heart attack?
Not necessarily. It's a decrease in sodium entering the cell to cause the neuron to fire. Okay? Basically it's saying you're stopping your depolarizations from occurring. That neuron can no longer ever fire again. So what they did with this pufferfish is, because as you know, these chefs spend a long time learning how to cut the fish appropriately to make sure that they're not severing any of the glands. Because if you sever any of the glands, well then that causes the poison to taint the fish, and anybody that eats it will pretty much die. Okay? Obviously they have their neurons stop firing. So they learned at the time when they were preparing this, and they saw people that ended up dying from eating fugu. And why did they eat fugu in the first place? There's enough toxin in the normal flesh that can be edible that it just causes like the slight numbing of the lips. And there's a certain excitement, to sort of like dance with death that people would have when they eat pufferfish. And they found that people, when they eat the pufferfish and it was a bad sample, well, they knew they died. And they found that at the time, in these old graves in Japan, they found the people that they buried due to fugu poisoning, they unearthed them and they found the bodies in weird configurations, and they found a bunch of scratches on the inside of the coffins, which means people were buried alive. So there could just be enough toxin that they may look dead, but they're not really fully dead. And after a while, our immune system can start breaking it down, we eliminate it, and then they restore back, and they found these people buried. So what they started doing with people with fugu poisoning, if they thought they were dead, well, they would actually have the grave, and they would leave the body right next to it. And they would just check after a couple days. If they noticed signs of decomposition, okay, person didn't make it. If the person's still the way they are, well, give them a couple more days and see if they wake them up. Otherwise, if they didn't make it, just roll them back in, cover them up, and that person's gone. So this video is it's a long video. You don't have to watch it, but I'll play it for you. It's only 3 minutes now?
This fish is
okay, well, now
delivered
you
to
can
the
watch
Tokyo
it.
market
I thought it was a
fully
10-minute
live.
video.
Although fugu fish lives in the ocean, hieroglyphs used for its name can be translated as river piglet. Looks funny and harmless, but terribly poisonous. When he meets enemy, his
really cute fish,
appearance
too.
turns threatening. In the ocean, it always helps.
They get
Fugu
threatened, they
has
blow
no
up, to make
enemies
themselves
there,
bigger.
but in Japan, nobody is scared. Annual sales of the fugu fish here are over 10,000 tons. Butchery is executed at special organized places. Way of cutting varies depending upon the particular species. To treat a fugu fish skillfully like this, a butcher has to study at least 2 years. Tetrodotoxin is a poison that has a nerve-paralyzing effect. It's concentrated in the intestines and even in the fish's skin. In case of exposure, effect starts in 10 minutes. Risk of fatal outcome is about 60%. The poison paralyzes muscular system and the man loses ability to breathe. Toxic rubbish is processed apart from regular one. Master is responsible for that. Even a drop of poison can be fatal. How to understand that poison is removed? Well, a processed fish has to be like this. All that's left here is inedible, but even after that, there has to be an additional treatment. Particularly this kind of fugu also has poison in its rear parts, says Master Yoshihiro Osada. Who was the first to taste the fugu? Nobody knows. At the end of 16th century, there was a special act of military governor of Japan prohibiting fugu fishing. Group of samurais couldn't survive after an evening party. After that, ban of fugu was removed and put into force several times. Today, fugu is acknowledged delicacy in Japan. It can be prepared only by licensed specialists. Each prefecture has its own regulations. In Tokyo, they were the most strictful. There is a special fugu department in Tokyo Metropolitan Office, but under the pressure from the customers, officials agreed to remove some of the limitations. Until recently, even after removal of poisonous parts, only specialists had the right to sell the fish. But reality shows that this kind of precautions is unnecessary. Now processed fugu can be bought from ordinary dealers, even through the internet, says Megumi Sato, head of the Food Sanitary Control Department at Tokyo office. Nevertheless, dozens become the victims of fugu in Japan every year. Almost all of them are amateurs who risk their lives trying to make a fugu sashimi by their own. Maybe now, when fugus become more available, number of those who want to play a Japanese roulette also becomes smaller. But there is one person in Japan for whom the taste of fugu remains a mystery forever. It's said that fugu probably the only dish of Japanese cuisine that can never be served to the Japanese emperor. Сергей Мингажев, Алексей Бичко, Вести, Tokyo, Japan.
Okay. So that, like I said, this is the puffer fish, the mode of action. This is the video. I think this was the long video. Maybe not a long video, but you guys can wrap up. You guys can see what's taking place on how to.
In persistent and chronic pain conditions, long-term changes occur in the functioning of pain nerve cells, making them highly sensitive and active, a phenomenon known as sensitization. The sodium channels located on nerve cells are in part responsible for sensitization. They act as gates that regulate the entry of sodium ions across the cell membrane and play a critical role in the transmission of pain and sensory signals to the brain. TTX reduces the activity of sensitized nerve cells by selectively and reversibly blocking these sodium channels. Although sodium channels are complex structures, TTX selectively binds to the fourth domain on the outer core of the channel. This blocks sodium ions from entering the nerve altogether, thereby interrupting or preventing nerve conduction. As a result, persistent and chronic pain may be relieved without the morphine-like side effects associated with existing pain management drugs.
That's really it. All right. Well, that's the end of today's lecture. I will see you on Thursday.
So-
What's happening on Thursday?
Quiz?
Quiz. All right. So study up. That's going to involve cellular respiration, the tissue lecture, and today's lecture. You guys have any questions? Feel free to hit me up. Otherwise, have a good night. I'll see you